So the objectives of NANODOPE are promote in situ monitoring of essential oceanographic variables from the different geographical locations of Asia, Africa, and Latin America. And we have also capacity building options, and we have also outreach activities. Currently, we have about 14 sampling sites, and our members are the representatives of these sampling sites. So they are responsible for monitoring the stations. And this webinar is a part of our NANODOPE activities. So in this webinar, we usually invite the members of NANO to share their research findings with us. So this is sort of capacity building option, so they can share their knowledge with us. And this webinar is open for everyone. So both members and non-members of NANO can join in this webinar. And today we have Dr. Arvind Singh as our speaker, and he is from Physical Research Laboratory, India. He will talk on effect of ocean acidification on marine nitrogen fixation. Dr. Arvind Singh is an associate professor at the Physical Research Laboratory, Ahmedabad, India. Dr. Arvind Singh is working in the field of ocean biogeochemistry. Broadly, he is interested in estimate the rates of diazotrophic nitrogen fixation and primary production using enriched isotopes of nitrogen and carbon in the Indian Ocean. His interests also lie in understanding the role of environmental parameters on the variability in the observed rates. His latest interests include understanding the variation in C&P ratio stoichiometry in the ocean. Currently, Arvind is the chair of the Early Career Network of the Second International Indian Ocean Expedition, editor of Journal of Geophysical Research, Oceans, and former member of the EZU Ocean Science Editorial Board. Arvind attended the 2011-2012 NF-POGO Center of Excellence in Observational Oceanography and is a member of the NF-POGO alumni for the ocean. So after this talk, there will be a question and answer session, but Arvind agreed that during his talk, if you have any question, you can ask him. So if you have any question during his talk, just click on the reaction button and then raise your hand. And for all of your information, this webinar will be recorded and then we will upload this in our POGO YouTube channel later. So if you have any reservation for being recorded, you can switch off your camera. Thank you. And thank you, Arvind, for accepting our invitation. And now it's over to you.
Okay, thank you, Subrata, for a nice introduction, and thank you, Lika, and also for having me here. It's a wonderful opportunity to share me the work that I have been doing with you all. Okay, I'm stopping my video, and I hope my screen is visible to everyone. Subrata, can you confirm?
Yes, it's working fine. Thank you.
So without further ado, let me start my presentation. This is on the effect of ocean acidification on marine nitrogen fixation. Also, if you have any questions, please do ask. I'm not sure if I will be able to see your hand raised, so you can just jump in when you feel like that you have any questions.
Shubrat and I will keep an eye on that. They can also use the chat and we interrupt you.
Okay, sure. Thank you. So this very simple carbon cycle slide that you might have seen before, but just for the sake of completion of this talk, I need to show it to you. So as we know that the atmospheric carbon dioxide has been rising for the last more than 150 years. Presently, it is more than, let me use my pointer. Okay. So these are the numbers in gigatons or petagram. The fluxes are in gigaton per year. The inventories are in gigatons. So presently the atmospheric carbon dioxide, as we know, it is already more than 400 ppm, and part of this atmospheric carbon dioxide also goes to the ocean, and part of this also comes back to the terrestrial system. So the numbers in red are the anthropogenic emissions, and as we can see here, it is from the anthropogenic activities from the land to the atmosphere, it is going off the rate of 7.4 gigaton per year. But in the atmosphere, it is increasing only by 3.4 gigaton per year. And this is primarily because whatever CO2 we are putting into the atmosphere, 25% is taken up by the ocean back, and then 25% is taken up by the terrestrial. But this whatever goes into the ocean, that comes there is some cost that we are paying, and we do not realize what it is. So for that, the concept of ocean acidification comes into the picture. For example, here you see the CO2 in the atmosphere, this seasonal variability of CO2 and rising up nicely. At the same time, this blue curve is the partial pressure of carbon dioxide in the seawater at a station in the Pacific Ocean. This also goes up and down. There is large seasonal variability because of the phytoplankton uptake and because of the change in seasons. And at the same time, we have the pH of the ocean going down. It has already gone down by roughly by 0.2 units, but this slide is slightly old, so the numbers are not revised. And how does it happen? So we know that CO2 is a gas. It dissolves in the seawater, and then you can call it CO2 aqueous. It reacts with water. It forms a carbonic acid, and carbonic acid is a weak acid. It dissociates further to form the protons and the bicarbonate ions. The ocean water carbonate chemistry is such that these carbonate ions, which are already there, they react with the protons that are produced, and they produce the bicarbonate. So in a nutshell, what happens that these black numbers are the pre-industrial evolution numbers. The numbers in red are the double of the pre-industrial numbers in terms of the CO2. But these are the projections for 2050. So I was muted. Were you able to hear me when I was speaking?
Yes, yes.
Okay. So these numbers in red, these are the projections for 2050. As we see that the CO2 in the atmosphere goes up, the carbonic acid and plus the CO2 aqueous also goes up, the bicarbonate goes up, but at the same time, the carbonate concentration goes down, and this is how the ocean carbonate chemistry works. The DIC total will, of course, will have to go up, but the pH also decreases. So the decrease in pH along with the decrease in carbonate ion concentration, together this whole thing is known as the ocean acidification, because we are decreasing the ocean pH. One important thing that we need to notice here is that the ocean pH will never go down below 7. It is just the trend of the pH, the decreasing trend of the pH, that is known as the ocean acidification. So the ocean, even if you put all the CO2 that we have in the atmosphere into the ocean, then also ocean will remain basic. The pH will remain above 7. But this may look like a very small number, 0.2 units, 0.1 units of pH, but this is quite huge because we are measuring it in the log scale. Okay, and some of the impacts that we already know of the high CO2, I will just like to recap, which you already know, which is quite a bit talked about. We see that it directly influences not just the atmospheric temperature, which we know as the global warming, but because ocean also absorbs heat, so the sea surface temperature also rises. The sea level, because of the thermal expansion, sea level rises. Ocean oxygen minimum zone also expands because the ocean will lose also the gases. The stratification, because of the less density at the surface because of the warming of the ocean. There can be also a change in nutrient stoichiometry and the sea surface salinity, increased precipitation. So these are all, there can be also some other parameters which probably we have not discovered that there is too much because of we are putting so much of CO2 into the atmosphere. Then can be too much influence on the other parameters as well, which has not been discussed yet. But these are all indirect influence, and many of the climate skeptics, they may not even believe that this can happen, or they would like to rebut that these changes can also happen because of changes in solar forcing or changes in any other parameters. But the rise in atmospheric CO2, it directly influences the ocean pH as well as the carbon species distribution. So there is no dispute about this. There is no other way through which we can explain the decrease in ocean pH, and that is why it is quite important. Unfortunately, this is not a much discussed topic. I mean, of course, it is discussed topic, but as this has not gained as much attention as the global warming has got, and these are two completely separate things. The cause could be the same, and this together, as I said, called ocean acidification. So my research in the last 4-5 years has been to understand the impact of this ocean acidification. You can also call it impact of high CO2, and how does it affect the ocean nitrogen cycle, and how do we study it? And the question is that why should we care about nitrogen cycle? And then who's getting impacted? So just I wanted to highlight that these, of course, these are beautiful looking corals and coccolithophores, and can also be some other calcifying organisms which have been studied from the ocean acidification point of view. But not only these beautiful things, there are also some other uh, players in the biogeochemical cycling that might be getting impacted, but we do not really know that how they are getting impacted, and that's have been the aim of my study. For example, these are the nitrogen fixers. On the left side you have the free living, on the right side you have the diazotropes, which forms kind of symbiosis with other bacterioplankton or with other plankton, and these are the very well known nitrogen fixers that we know right now, and they can be impacted very differently, and that is what we do not know. I want to take this out. Okay, so why is nitrogen, I mean, why should we worry about the nitrogen cycle from the ocean acidification point of view? Because nitrogen is the most important, and it is the ultimate limiting nutrient in the ocean that affects the oceanic health from the primary productivity point of view, and even from the carbon sequestration point of view as well. And another misleading concept that we know, I would like to rebut that here. You know, we, in the slide of the carbon cycle that I showed, a lot of people attribute this additional CO2 removal by ocean to the phytoplankton. So that is not true. The ocean phytoplankton cycle, or the biological pump, that seems to be in the steady state. So although we are putting more CO2 into the atmosphere, the activity of phytoplankton, more or less, it remains the same. It is just the physical activity that actually has changed, because we have more CO2 into the atmosphere. Because of the difference in the partial pressure between the atmospheric CO2 and the ocean CO2, there is more pumping of CO2 into the ocean. So kind of solubility pump. If CO2 was a limiting substrate, then that could have been true. So we don't have very strong evidence suggesting that the phytoplankton activity or the carbon uptake by the phytoplankton that has changed over time because of the high CO2 into the atmosphere. But the nitrogen, but is still on the geological time scale, we know this biological pump is important. And if we put additional nitrogen inputs into the ocean, then this biological pump becomes important. And likewise, if you take nitrogen from the system, nitrogen from the oceanic system, then also this biological pump would become important. So any changes in the nitrogen cycle will ultimately affect the ocean biological pump, and hence it is important to understand it. So like nitrogen fix, and my focus has been only to understand the nitrogen fixation, which is one of the most important process through which the ocean receives nitrogen. And although there is plenty of nitrogen in the atmosphere, 78% is N2, but that form of N2 is not bioavailable. Most of the phytoplankton, they cannot utilize this nitrogen, this form of nitrogen, the N2, because of the strong triple bond of N2 that requires a lot of energy to break it. But there are these cyanobacteria, we call them diazotrophic cyanobacteria, which can actually break this one. And my focus has been to understand that how ocean acidification will impact these cyanobacteria and how will it change the oceanic nitrogen fixation. And I'm not the first one who is doing this. There have been a lot of studies to understand the impact of ocean acidification on nitrogen fixation. But if you just read over only the title of these papers, you will see that these are the results, the conclusions are very different from one to each other. Some of these results show that the ocean acidification will negatively impact the cyanobacteria. That means the nitrogen fixation will decrease. Some suggest the nitrogen fixation will increase. And the problem is that these contradictory results, and the reason is that the conditions and the species are different. For example, the slide I showed that these different kind of cyanobacteria, they have a different niche. They like different kind of environment. So the effect of ocean acidification will also be different on different species. And most of the studies done until 3-4 years before, they were done in culture experiments. So like in very small bottles in the lab. So the community level experiments were not there, and that could have been the reason. And once the ocean acidification affects the ocean, it will affect on the community level, or rather on the oceanic level, or at least on the regime level. So we need to have that kind of understanding. And that is the reason we went to do this experiment, the results that I will show in there. And the hypothesis from our study was that the future PCO2 levels, that means the future high atmospheric CO2 levels, will increase the N2 fixation rates. And the reason behind this hypothesis is that this phytoplankton or the cyanobacteria, because of the availability of high CO2, they can utilize this energy not to fix this CO2, which is already available, but rather they can utilize this energy to fix nitrogen. That was the thing. Additionally, in this experiment that we did, we also added the deep water into our mesocosms. And our hypothesis was that the addition of deep water will decrease the N2 fixation. And the reason is because the deep water has already plenty of nutrients. When the nutrients or the nitrogen nutrients are already there in the system, why should the cyanobacteria need to fix N2, which will require a lot more energy? So with these two, and this is the design of the mesocosm setup. So we did this experiment in Gran Canaria in 2014. So one of these Canary Islands in the Gando Bay. And this is how setup of our mesocosms were. And how would the mesocosm look like? So we had 9 mesocosms. These are made of 1 millimeter thick polyethylene material, 2 meter diameter, and they can take around 50,000 of liter water in there. And the PCO2 was manipulated between 400 micron atmosphere, which is present day partial pressure of CO2 in the ocean, and also the future looking at how would the oceanic PCO2 would be in 2100. So this was our target, and the experimental length was up to 8 weeks. During September to November 2014.
Arvind.
Yes.
Can I ask you, how do you control PCO2 in these mesocosms?
Okay, so I think in this slide, that's a very good question. So we don't really control. We put the enriched CO2 into these mesocosms, and then of course, because they are supposed to mimic the natural conditions as natural as possible. So like these, if you see the top of it, they are open to the atmosphere. But once you put high CO2 into the ocean water, you will form the carbonic acid, and further it will dissociate to bicarbonate and to carbonate. So the CO2 as such in the aqueous form will remain low, but it will shift because the pH will also shift as a result of high CO2. That will also shift. So there will be more DIC, not as, I mean, the PCO2 may not be still very high, but also because we are leaving it open to the atmosphere, it will diffuse back to the atmosphere. So what we do, we will keep it, I mean, after a period of 10 to 12 days, the CO2 level will decrease down rapidly, and then we will again put the CO2, we will try to get it back to the original level. So the PCO2 as such keeps on decreasing. Even the DIC keeps on decreasing over time, but then we like top up after some time if we have a long-term experiment. Because if you want to control, if you want to lock it up, then there may be some other practical, difficulties to mimic the natural conditions. It will heat it up, and there can be a lot of other problems. So that result I have, you will see that how the DIC goes up and DIC goes down. Is it
Okay,
okay?
thank you.
Yeah. So this was like a 58 days experiment. So 55 minus 3, or rather you need to add these 3. And we start our experiment at the 0 time when we add the CO2. So this is the CO2 addition. And the CO2 also added somewhere in between, but I forgot to mention it in this slide. I think 2 times or 3 times it was added over the total experimental duration. And we did the phototrophic nitrogen fixation experiment as well as the chemotrophic, or I don't know whether heterotrophic, because they were done in the dark, so they can still be chemoautotrophic or can also be heterotrophic nitrogen fixation simulations. So I am showing only one.
Arvind,
Yes.
there is a question from Shudesh.
Yes.
Do you measure the carbon dioxide in between?
Yes, we measure every day, or rather every other day. We do the sampling every other day. So we in each mesocosm we measure. So those results are there. Yeah. So okay, and this is, I am only showing 1 or rather 1% or 5% of the work done in this mesocosm. There were a lot of other activities, and my focus was only on the nitrogen fixation. So that of course you can't set up such a big experiment only to do the nitrogen fixation activity experiments. And these are some of the other results. If you want to see that the CO2 levels and how much was the volume of water in each mesocosm. So these are M1 to M9. M1 and M9 were controlled, so no CO2 was added in this. And then we added the CO2 in each of the other mesocosm from a regression level, not from the ANOVA point of view. So there were no replicates, but rather increasing trend of CO2 we added. And in the, okay, so there were 3 different phases. Like first phase was the oligotrophic phase, and then in the phase 2, after we added these deep water nutrients into the mesocosms, and then the phase 3 was when the nutrients were consumed up. And then this is the PCO2 in micron atmosphere in the each mesocosms that you see here. Of course, our target was 1400, but we can never reach to, I mean, it is difficult to reach that target, because there will all likely cast, how do you maintain it? So we can't maintain it. So there will be diffusion of, or the CO2 will be diffused back from this mesocosm to the atmosphere. So you can see there is in different phases, there is the change in the PCO2 concentration. And this is the mean level of PCO2 concentration. Of course, there were also trouble. There was a hole in M4 mesocosm, and somehow we also, because there were strong currents one of these days, so one of the mesocosms we lost, and also there was hole in the M9 mesocosm. But these holes we were able to fix. You can also ask interesting question: how did we get to know because there is hole? So that I can answer later. And this is the sampling for N2 fixation. So we would go like every other day to these mesocosm and take these using this boat from the lab in Cloklon, which are nearby. In this boat, we will go and take the samples using this integrated Niskin water sampler. So for gas sampling, this is the preferred one. Otherwise, if you are not interested in the gas sampling, then there are also other ways to take the sample. And then we will put this in the Nalgene bottles. We will transfer. bring them to the lab in Plocon, in Gran Canaria, and do this, put the enriched tracer of 15N2, and then incubate it like we do this on the ship-based experiment, and then filter them out. Just one slide on the deep water collection, because this was also very intriguing. Of course, I wasn't there on the ship at that time, but this was quite interesting that we were able to collect the deep water, quite a lot of thousands of deep water from the 800 meters from the ships, and then we put it back into the each mesocosms. And if you want to know the amount, this is the amount of deep water that we added in these M1 to M9. So of course, roughly like 20% of the water we replaced, or rather we did not replace because there was already continuous evaporation. So we just topped it up with the deep water. And these are the nutrient results. So as you can see, these are the different mesocosms. And here by M1 to M9, I have replaced by the PCO2 levels, because that is more meaningful. So the blue ones are like the low PCO2, red ones are the high PCO2. And you have also the Atlantic water we took, and this is the Atlantic water that you see here. So Atlantic is like real, like control. These ones would be control, and this is like control of controls, because the mesocosm will still be isolated from the ocean water. So this is nitrate plus nitrite, this is phosphate, this is silicate, and this is P star, which is nothing but phosphate minus nitrate divided by 16. So just a measure of excess phosphate from the Redfield ratio point of view. And you can see that this nutrient concentration goes up, and this goes up because we added the deep water. So this goes up from almost 0 micromole per liter to 3 micromole per liter. So this is a huge amount of deep sea water that we added. This is what happens. At the same time, the P star goes down because there was excess nitrate in the ocean water, and this is quite important to explain our results. So you may need to keep this in mind while I explain the results.
Sorry, Arvind, it got increased at day 20, 30 because you added deep water?
Yes, because we added deep water into the mesocosms. So since deep water has nutrients, the surface water doesn't have much nutrients, right? Very close to detection limit. And deep water is nutrient rich. So we added this nutrient rich water to the mesocosms, and that is why in the mesocosms the nutrient concentration also increased. But this all the nutrients were consumed in 5 to 6 days. So this was like our oligotrophic phase. This was our growth phase, and this is our decay phase that I
and it's the same water in all experiments.
Yes, in all, okay. except the Atlantic, you see the increase everywhere.
Yeah.
So in each mesocosm we added, and these are the PCO2 levels that you may need to keep in mind. And this is the thing, like we are not able to maintain the CO2 levels. So because of the diffusion of the CO2 from the ocean to water, the CO2 would, of course, initially it would increase, and then it will go down. Then we put up more CO2, then it will rise up, then it will again go down. And this could be because of the deep water, because we added a lot more CO2 in the mesocosms. Deep water doesn't have that much of carbon, right? So, and then again it will go up. So similarly for PCO2, this is the pH if you want to see that how much it decreased. This is the temperature and salinity variation. Not very important to explain my results, but if you're interested that the temperature went down, this is just the natural temperature that went down because we started somewhere in September and this was already mid-November or so. And the salinity would go up because there is evaporation. These are like a closed system. This mesocosm is like a closed system. So there will be evaporation and it will increase the salinity. So the salinity increase up. But when we added the deep water, this would go down. This is the AOD, the aerosol optical depth, the dust AOD as well as the total AOD. Dust AOD is a measure of atmospheric dust. Now, why I am bringing the atmospheric dust here? Because dust brings nutrients to the ocean, and mainly for the nitrogen fixation, iron is one of the important nutrient that the dust brings. So we also wanted to see the natural activity. So there were 3 dust events, like first this one, then just before the second phase, and then at the start of the third phase, we had these dust events. So these were also there. And these are the main results of my work. So we have on the y-axis the nitrogen fixation rates in the light incubations, in the dark incubations, and then the carbon fixation rates in the light as well as in the dark incubations. And then after the, so this is my deep water addition here, where this phase starts. And again, the blue ones are low CO2, red ones are high CO2. So you can see the nitrogen fixation increases, but it doesn't increase immediately after the deep water addition. It takes time. In fact, when all the nutrients are consumed up in this phase, then it increases even further. And when the CO2 is high, then the N2 fixation is even high, which is quite true for the light. In the dark one also, it was quite a bit similar, but the effect was not very prominent to see because the rates were quite low. In the carbon fixation rates or the primary productivity, it increased immediately because the system was nitrogen limited. So it increased immediately after this deep water addition and then went down. And then also in the high CO2 here somewhere, in the high CO2, the carbon fixation was high. So from these results, it seems like that the high CO2 kind of enhances nitrogen fixation as well as carbon fixation, but only when the nutrients were plenty. So like in the decay phase only it increased. So perhaps this phase could be when there is a lot of competition. There were other non-diazotrophic organisms were here, and the diazotrophic organisms were outcompeted, so they grew a little later. So that could be the reason, and that can also be seen here that I have put a question mark there. The addition of deep water decreases nitrogen fixation rates because this was our hypothesis. So of course it doesn't decrease, rather it increases afterwards. If it was decreasing, it would decrease here, so it doesn't decrease. And also, other than the iron, the phosphate is the other important nutrient for nitrogen fixation. Where there is plenty of phosphate, plenty of iron, then the nitrogen fixation is likely to happen. So the N of phosphate, but no increase in nitrogen fixation rates. So here you have the phosphate. So there is no increase in nitrogen fixation here. One thing I forgot to mention, I should mention that if you see the Atlantic, in Atlantic we did not put anything, we did not manipulate anything, but the Atlantic rates were also increasing. So our kind of control rates were also increasing, and this we attributed to those dust events that I showed here. So perhaps the dust events also played a role that we cannot take out from this, that this is only the addition of deep water that increased this, but the deep water will also bring the iron nutrients as well, that will also increase nitrogen fixation. Okay, and the nutrient stoichiometry plays a role. So again, if you look at the P star values, this is excess phosphate. So let me take this out here. Okay, so when the P star was negative, then the nitrogen fixation rates were still low. When the P star values return to original or somewhat positive values, then the N2 fixation increases. So the point that I want to make from this slide is that it is not the absolute phosphate that matters, rather it is the excess phosphate that matters, and that is from the competition of the different organisms we can explain. Okay, and this slide has the diazotrophic abundance. So I have the UC by unicellular cyanobacteria. These are all nitrogen fixers. So like the random going up and down, and the UCY and B showed rather quite interesting feature that it increased after the deep water addition. The gamma protobacteria as well. Probably these were when we put the deep water into the mesocosms. Probably these came from the deep water. So our nitrogen fixation rates matches quite well with the UCY and Y and B. So it seems in the high PCO2 world, probably these unicellular cyanobacteria are going to flourish more, and they will bring a lot more nitrogen from the atmosphere or other fixed atmospheric nitrogen into the ocean. So they will help us in many ways.
Arvind, what's the difference between these two cyanobacteria, A and B?
These I mean, I don't know quite well about them, but if I can show you that slide. So from the symbiosis point of view, they are different. Also, their activity is is quite different. So like UC by Na you have here, this is also known as UC by Nb. So from the symbiosis point of view, they are different. They fix energy or they fix N2 from very different ways. Some of them fix in the night. Some of them do it together with carbon fixation during the daytime. So that is the that is broadly that is the difference in between them.
Okay.
Yeah, and these
Another.
are all, and until now these are difficult to culture, so we don't really know too much about them. Yeah. So if I can summarize, then the future PCO2 levels will increase N2 fixation rates, provided there is sufficient phosphorus and iron. So these results, although when I compare with the literature results, they somehow match with them. They are not completely out of line with those results. If you put all the results together, then they tell almost a similar story that for nitrogen fixation to increase, alone PCO2 will not do anything. You need to have enough iron and enough phosphorus. And then the addition of deep water, all the future, if the future of whaling events will increase, and some places they will decrease whether they will decrease the nitrogen fixation rate. So that was our hypothesis, and this kind of we have disproved that it is not likely to decrease the N2 fixation rates, and it is not the absolute phosphate that matters, rather it is the excess phosphate that matters from the competition point of view. And these are the, because this was a big teamwork, one person or a handful of people cannot do this work. So I would like to acknowledge the Cosmos team and the Gran Canaria GC 2.0 participants, because this was the second mesocosm we did in Gran Canaria. And then of course Plocon for all the facilities they have provided in Gran Canaria, Future Ocean for funding my postdoc, and of course GeoMarkeel for everything. So I would like to stop here, and I hope I was able to explain. I was not in too hurry.
Thank you, Arvind, for your very informative talk. So now is the time for the question and answer session. So participants, if you have any question, you can just
Suji. Yeah, Suji has a question in the chat. Maybe he can join us.
Yeah,
Sudesh.
Thank you, Lika. Arvind, it was very nice presentation. I'm very much happy to see the experimental setup. Like it was definitely, there is no doubt it would be a wonderful experience dealing with that such a like a very in the in situ experiment. So my question is like when you add the deep water into the system, into your mesocosm, you are telling like there are some fixers, they are more active. So is like mean the fixers living in dark, when they may be more active when they enters into the light condition. So is it can be attributed like that?
I think it will be difficult to answer, and I don't think, I mean, I mean, I can only guess. I cannot give a certain answer because we do not know. But I guess since they are in the dark, there is a reason that they are in the dark. They like to be in the dark. So
because when I say the Atlantic also more, your control also has some good changes. So
yes. So the Atlantic be attributed to this dust event. There was dust event, which is fortunate or unfortunate, I can't say, because if there was no dust event, then whatever our results were, we could have attributed to deep water upwelling. But since we don't have a control over our control somehow, so we can't really say. But it looks like that is because of the dust events. And since our Gran Canaria is very close to the Saharan desert region, so these dust events are quite frequent there. So that's a natural thing, yes.
Yeah, because like if you see our Arabian Sea scenario in the northeastern Arabian Sea, all these things happen naturally because the convective mixing will happen. Also, the dust is event like because of the winter cooling, like the winds will bring more dust. So these can be more like natural in our environment. That's why I asked. And I also have a question from one of my reviewer. One of the reviewer, when I submitted to one OMSAT paper to Professor Oceanography, that like this is the same question, whether the bacteria, because there I saw some interesting feature that when there is a convective mixing process happen, the surface oxygen going down because of the definitely the low oxygen waters are mixing with the surface waters. So the question was, when there is a like a deep water mixing to the surface, whether the bacteria in the deep, whether they are more active in the light condition. Such question I the reviewer asked. So the same way, like when I see your result, I was thinking about that.
Okay, very good. Yeah, so Sudhesh, I completely agree. I think in Arabian Sea, I mean, whatever I did in these mesocosm, we have like a natural laboratory existing in the Arabian Sea, and perhaps this is the reason we get very high nitrogen fixation rates in the Arabian Sea. That could be the reason. And about your second part that you are dealing with the reviewer, I have just one comment. So these nitrogen fixers, they are also sensitive to oxygen. When the oxygen is low, probably they will fix more nitrogen. But somehow their enzyme setup is such that they are able to manipulate this oxygen. Even if when the oxygen is high, they will make kind of a virtual shell or some shell, and the oxygen will be away, and they can still fix N2. But when the oxygen is not in the environment, perhaps they would be even more happy to fix more N2. So, but this is only for the nitrogen fixers point of view.
Okay, thank you. That will be more interesting, I think.
I have one question, actually more of a curiosity. I also noticed that Canarian is very close to the area of the Sahara Desert. And was that intentional, that the decision of putting the mesocosms there?
So, I mean, so before this mesocosm study, so this is GC 2.0. GC 1.0 was done in the same year, but that was in winter, I think. And before that, whatever studies were done in these mesocosms, they were all done in the polar regions, in Arctic and all. You might have seen the slide. So we wanted to see, and most of the ocean is actually the oligotrophic ocean is actually in the tropical, subtropical, or temperate regions. So my postdoc advisor or his team, they all wanted to do this first open ocean. I mean, this is still this is very close to land, of course, but kind of simulates the open ocean oligotrophic conditions. So this is what we wanted to test there. Close to Saharan or not, that wasn't intentional. It was just the opportunity where it was available. At that time, it was available in the Gran Canaria. They were happy to host us. So we went there. But I know after I left Geomar, they also went to do several mesocosms in Peru as well, in Peru or Upwelling regions.
On Upwelling region.
After that.
Oh,
yeah.
Interesting. Yeah, and your result can be summarized that the nitrogen fixation can increase as long as you have enough phosphorus. Yeah.
Yes.
What is the main,
as well as iron. Yes.
And iron. Yeah. What is the main sources of phosphorus?
What is that?
The main source.
Okay, so the main source is from the terrestrial inputs. It doesn't come much from the atmosphere. There is not much atmospheric transport. So mainly from the riverine inputs, and riverine inputs are mostly consumed within the estuarine region. So they also don't reach to the open ocean. So the only now source remains the upwelling. So perhaps in the upwelling regions, they will be, if the CO2 is going to increase in future, so in the upwelling regions, probably N2 fixation will be more. This is what we can say from this study.
Yeah. So just out of curiosity, I just Google it. What would happen with the Sahara dust with climate change? And I found a link from NASA saying that the dust plumes might actually shrink with global warming. So, and this is one of the iron sources, right? So maybe the system find a way to control and balance it out.
Yeah, I mean, we don't really understand the nature. I mean, so this is somewhat we try to mimic nature as much as possible. Like the other experiments, they were all in the labs. So this was quite good in many ways. but still you can never mimic the nature. They're like ocean physics, you can never mimic
That's
in the
true.
ocean.
But
Yeah.
still,
Yeah.
it was very good exercise.
Yeah.
Arvind, like
Yeah.
in your one of your slide, you have mentioned about the hole. Like,
Yeah.
how did you identify this? Because
Okay. So, yeah, that's good
you
that you
told
asked.
me.
So like when you have the closed system, then the evaporation continuously happens, right? So the salinity was going up. If I can, let me see if I can have that slide. I think so salinity, right? So so the salinity keeps on going continuously because it is a closed thing. Although it is in the ocean, but it is locked. So the ocean water doesn't go in the mesocosm. And when there is a hole, the ocean water will also go in this.
Okay.
So the salinity will not change. So we found that the salinity wasn't changing in these two mesocosms for some time. Or the salinity in the beginning it was increasing and suddenly it changed. For example, I think here in M6 you see this this went down. Uh, or maybe somewhere earlier it went down somewhere. So when the salinity suddenly went down, so it cannot suddenly go up or it cannot suddenly go down when it is a closed system. So from the salinity observations, one could identify.
Okay. Okay. Nice.
Mm.
That is interesting.
We still have time to take one or two questions. So if you have any questions, please feel free to ask.
What time of the year this experiment was done, Arvind?
This was September to November
2014.
2014. Yes. And these results, I mean, I took a lot of time to write because at the same time I got a job in India. So the results were published this year only in limnology and oceanography. I think in the
Is it
first
already
month.
published?
Yeah, it is published.
Okay. Okay. Then I'll search.
So it is in limnology and oceanography this year we published.
Okay, because one of our recent paper we are doing the excess phosphate from Arabian Sea. Like so the same like as you said, when there is a high like increasing the stratification because of the increase in sea surface temperature, definitely this deep mixing won't happen as expected, like as usual. So in such condition, do you expect more nitrogen fixers? Like we have already Arabian Sea has excess phosphate compared to other oceans. So how do you think? Like suppose if there is no dust or if there is any dust condition.
I think as per our traditional knowledge of nitrogen fixers, we expect more nitrogen fixation when there is a stratification and warming of the ocean. Uh, but I mean, our most of knowledge is trichodesmium centric, and now we have these new players, unicellular cyanobacteria and all. We do not know much about them, much conditions that what kind of conditions they like. So and now I mean, they are kind of everywhere. Like trichodesmium, they are in some places, some hot spots, and these were are kind of everywhere. So it's hard to see, but our traditional knowledge point of view, I would say the nitrogen fixation will increase in future.
And Aravind, is there any low-cost options to set up such type of experiments?
I didn't get Subrata. Can you repeat?
Is there any low-cost options to set up such type of mesocosm experiments?
So as far as I know, only there are one or two groups. I mean, this is one group that I know that they are doing this in situ mesocosms. I don't think, I mean, it not only the cost, also there are a lot of manpower, a lot of expertise are needed for such work. So unless you get and trained in such a group, it won't be possible to do. For example, I have never thought about doing this in India because I knew that there is so much we can't be able to afford. But for example, we can do the lab mesocosm thing would rather be cheaper. So to begin with, to start it in the lab, we can do lab mesocosms, and they would be, I mean, they are low cost. They are not. You don't need to put a lot of money to do the lab mesocosms.
Were you involved in this in the cannery? Were you going out every day to collect the samples? Yeah.
Let me recall. So not every day. I had like 4 or 5 times. So we had like, you know, not everyone has to go on these boats.
Mm.
There was limited people that can go, and then how much amount you need. So if I go, I mean, everyone would do sampling for everyone. So like those who go on the boat, they will do sampling for everyone. So I think I went twice or thrice for the sampling. But otherwise, people like every alternate day, people will go to collect the samples.
Very nice.
So if there is no more questions, then I think we can close our webinar. I think there's no questions. So thank you, Arvind, and thank you for accepting our invitation, and thank you for your nice talks. And I also thank you all for joining us today. Thank you.